Today I am going to discuss about the cell culture techniques. So in this topic, the name itself it is implying that the culturing of the cells in the in vitro condition. So we should know about the difference between in vivo and in vitro system. And also about the cell. What is a cell? Cell is the structure and basic unit of one particular system. So it is the one which will be transferring all the signals to another cell, and it is the one which will be having the varieties of receptors that can able to transfer the signals. So these are all the basic functions which has to be adapted, and they can form the respective tissues, and these tissues can form the covering areas of the respective organs, and they are performing their fine functions. So varieties of tissues are available in our system. So all these epithelial tissues, for example, an epithelial tissue, connective tissue, all these tissues are present in our system. So all these tissues are made up of the respective cells, different varieties of the cells, so which are present along our system. So now we are going to cultivate, or we are going to grow these cells inside the in vitro, that is nothing but inside the laboratory condition. So how to perform that? So what all the techniques which has to be adapted, and also how to differentiate the in vivo cells and in vitro cells. So what all the majority of the functions are shared, or functions are similar to the in vivo or in vitro, that has to be discussed in this particular topic. So before going to see about in detail about this one, so this particular technique which was 100 years back developed one, and varieties of techniques are adapted, varieties of application emerged out of this particular technique. So the history of this particular tissue culture technique which was emerged by the Harrison in the year of 1907 start different types of cell culture which has to be maintained in the in vitro condition. Now we have to see about the different types of cells. So I already told you the cell is a structure and basic unit of one system. Now we have to see about the different types of cells. The cells which are covering, we can call all those are the epithelial cells. So like this, wherever it is present on and doing a specific function, we can call the different types of tissue. Connective tissue is also there, and varieties of the lymphoblastic cells are there. So these all the cells are present in our system. So they are present inside the system. So all these cells are made up of respective tissues. So these tissues has to be taken out first. From that we have to convert this into cells, and the cells has to be grown in the in vitro condition. So why we are performing all these things means this will be utilized for the varieties of biochemical engineers, and also the biomedical engineers, and also some of the chemical people and environmental people, they can utilize this one. So that is what we are doing this particular cell culture techniques. So now we have to know about the different cells which are available in the in vitro condition. We have fine cells, one is called as Anchorage dependent cells. Anchorage dependent means the cells need some substratum. So before going to know about this attachment, so they are basically these cells, each one will be attached tightly with one another. So how it is performed means it can be done with the help of the material which is called as extracellular matrix proteins. They are called as collagenase, laminin, fibronectin, all those things which are tightly bounded with the cell and cell attachment. So we can call this is the cell-cell attachment, or with this only the cells can able to transfer their signals from one cell to another cells, and they can communicate, they can share all the things. So otherwise we can kill cell talking is also present over there. Why means they can perform their particular function of all these pharmacokinetic and dynamic action has to be perfectly performed over there. For that only this attachment and everything will be there. So this is the attachment can be performed by the cementing material which is called as extracellular matrix protein. They are tightly bonded over there. If you are talking about this Anchorage dependent and non-Anchorage dependent, so first one is Anchorage dependent means the cells has to be present on one matrix, and then only it has to start form their extracellular matrix protein, and they can attach each other. So to perform this, so those cells which recover the substratum, we can call all those are the Anchorage dependent cells. Another one is called as suspension cells, or the Anchorage independent cells. They never need any substratum. They will grow as a suspension. Example, blood cells. So they will just as such which will be present over there. And also some of the cancer cells, they will never need any substratum. They will be present as such. So they never need any substratum. That is called as Anchorage independent cells, or we can call it as a suspension cells. So these are all the varieties of cells which are available in our in vitro condition system. Now we should know about that how they are the cell which is present on the in vivo will be same as in vitro. That has to be discussed. Our aim is we have to get like this only, then only our work is very good. So in that condition, structurally and functionally they have to perform whatever it is present inside the in vivo system. So if you are adapting that, our work is very fine and it is a good one. Now To achieve all these things, we need a varieties of instruments and also varieties of techniques that has to be adapted perfectly in our laboratory. To get a laboratory, animal cell culture laboratory, it is a very toughest job, because all the instruments and all the recovered elements, all the recovered medium and growth factors, whatever we are using for this one, everything is high cost. Nothing is cost effective, everything is high cost. So we need a high amount of money out of it, then only we can construct one particular lab. So in this lab we need a set of instruments which are used. So first one is we have to use a sterilization technique. So why I'm interestingly saying about this one means this sterilization is the first and the foremost technique which is utilized here, because in rest of the labs we are not using sterile environment. Here we should use the sterile environment. So the place also should be very contaminant free, and also the instruments, and also whatever the utensils or the whatever the material or the whatever the pipettes or whatever the things which are used, sterilized well. Then for that we are using two instruments, one is called as hot air oven and another one is called as autoclave. So these two instruments which will which is giving the contaminant free environment to the particular things, whatever we are using for the respective cell culture techniques. Next one is varieties of instruments. The first one is laminar hood. Laminar hood is nothing but the place where we are sitting and performing the experiment. So in this laminar hood has to be totally, that particular environment will be free of contaminants. If any contaminants is there, any microbes are there, so that will fully spoil our system. So that has to be adopted. So that has to be regularly fumigated, and it was totally sterile over there, and then only we have to start do our work. Second one is Co2 incubator. So the place where you are performing all the work, like we are going for any conversion of the tissues to the cells, or the cell has to be fed with the material, everything will be done in the laminar hood, because we have to prepare the growth media and it has to be present inside the laminar hood. Whenever we want, we will be added over there, and we will be preparing a fine work table is this one. So followed by we have to go and keep all the cells with the respective media and all the factors and into the respective Co2 incubator. This incubator where we will be storing, or otherwise we can say where it will be present into a home, just like a home, it will be sitting it over there and grow. This growth has to be perfectly maintained by the addition of all this recovered amount of the media and the sera, which is giving the strength to the cells, and they will start divide. So the division can be performed inside the Co2 incubator. Followed by, we will discuss in later about all these techniques and everything, what all the mechanism of action and everything. So this is the incubator where the cells are simply sitting it over there and they will start divided over there. Next one is we have to view the cells regularly. That is nothing but we have to, one day if you are doing the subculture, and we have to see the cells on the next day. So the viewing pattern, nothing but whether they are live or dead in condition, that has to be adopted by using a varieties of microscope. Here we have to use a inverted microscope. The light is coming from the top to down. So rest of the microscope it is reverse. So it is called as inverse. Otherwise we can call this is the light microscope or inverted microscope. Inverted means the light is coming from the top, and we can able to magnify our cells of interest which are present in the flask, and we can easily view that one. That is called the inverted microscope, or other name for this is light microscope. Then we have if we are having plenty number of cells in our hand, we have to go for freezing. Freezing is nothing but we have to store the cells. So the storing can be done with the help of the very good media that is called as DMSO, dimethyl sulfoxide. So to that we have to add it over there. The cell has to be added over there in recovered proportions, and we have to go for the slow freezing technology to store the cells. So these are all the necessary basic instruments, all are high cost, but these are all the things which are very important one for constructing one particular cell culture laboratory. Now we should discuss about the primary cell culture. Primary cell culture is nothing but we are converting we are taking the cells from the one particular tissue mass, or which is a covered one, covering organ one, which has to be taken out, and we have to convert this into the respective cell mass. So that is what our aim. In this case, now we have to do some more steps. So in the first step, we have to isolate a particular fibrous tissue mass from the one particular organ, and then we have to treat that. So treatment can be done by the primary treatment can be done with the help of after we are isolating this particular tissue mass, we have to wash it with a plenty amount of the media. Then we have to add the respective physical methodology has to be adopted, like we have to use a scissor and forceps to mince the cells into small pieces. Followed by we have to add chemicals like trypsin and collagenase. So they are the one which will be cutting over there that cementing material of the extracellular matrix, and also the attachment has to be totally, it was coming out. Then followed by the third one is we are also using the some instrument, that is using a magnetic stirrer. We have to put a pellet over there and we have to convert this cell mass, tissue mass into the cell mass. That is what our aim. So we are converting that into this, and the tissue mass into the cell mass, and finally we will be having a fine cell pellet will be on our hand. So this cell pellet has to be added with the recovered amount of the media followed by your growth factor. Then only the cell will start divide. So after this was perfectly done, we have to keep it inside into the incubator. So inside the incubator, it has to be start, divide, and you will be getting the full monolayer on the very next day. So this is what the regular protocol which is to be adapted. We have to know about media and serum. So varieties of commercial available media are presented over there. So we can get it from the high media. So many company people are giving this one. So commercial available media that has to be prepared perfectly in our laboratory, in the respective laboratories. Then followed by the sera, which is also commercially available from the so many people like Serotech, and also from the Jibco or from the high media. So those people will be commercially giving this particular varieties of serums. So what is the use of this media? What is the use of this serum? Media which is perfectly maintaining the pH inside the hood. Which next one is the cell has to be divided. For the division we need a growth factors that is present on the serum. The serum which is also a commercially available one. So these two are the important factors to grow the cells inside the laboratory condition. Then we have to, this will be varied depending upon the cell which we are utilizing, or cell which we are getting it over there, and we are finally maintaining it over there in your respective labs. This is what the primary cell culture. Suppose you are getting the cell culture from cells from the another procuring from the any other bank like NCCS Pune, National Cell Centers. So they are having the different types of the cells. If you are procuring from that area, so we have to go for maintaining the particular cell lines. So how to maintain this? So maintenance is another second technique. So in this we are going to see about the cell full monolayer architecture in the microscope. After that we have to remove the supernatant, or we can say that the tissue culture supernatant has to be removed over there. Then add about 1 ml of trypsin, versin, glucose. So this is called as TBVG. So what are the functions of these things means trypsin, which will be able to chelate the cell, that is wherever the protein attachment is there, or that will be totally cleaved over there. Next one is EDTA. EDTA is nothing but it will totally remove the wherever any ionic interactions are presented over there, and the glucose which will produce any osmotic effect and remove the cell attachment. So for that only we are adding this trypsin, versin, glucose or TBVG. So it is prepared along with the phosphate buffer saline. So this has to be added over there for about 1 ml into the cell wherever it is present on the matrix, and we have to wait for about 1 minute because this trypsin is toxic to cells. So remove it immediately. We are adding 5 ml of media, then we have to go for slow flushing. Slow flushing is other name for this is called as trituration. So slowly we have to flush the cells, and we have to transfer this cell into the 20 ml of media, respective medias which are utilized over there, and we have to segregate into 4 different flasks. So that is what the conversion of 1 flask into 4 flasks, and otherwise we can maintaining this particular cell lines. Our subculturing and maintenance can be performed correct way. So this is what the maintenance, subculture and maintenance of the particular cell lines. So why we are doing this one? So if you are maintaining only, this can be utilized for the research papers. Those who are coming for the research work, they can utilize it over there. So in that condition, after you are having plenty number of cells in your hand, so what you have to do means we have to store it over there. So storage can be done with the help of the special media that is called as DMSO. That is called as dimethyl sulfoxide. So we have to go for a subculture, take out the pellet of the cells. To that we have to add the respective quantity of the DMSO, like 10% of the DMSO along with the media that should be added over there, and we have to be keep it in the go for a slow freezing technology. So how to go for means we have to keep it in the fridge for some times, and then we have to go for a minus 120 degrees Celsius, and finally on the very next day we have to keep it on the minus 140 degrees Celsius of the liquid nitrogen container, which is called as cryo container, or we can call that as a cryo can. So we have to store it over there. So this is what the fine preparation of the cells, primary culture, and also the subculturing and maintaining the cells. So we have to go for a cell counting. How to perform the cell counting? So we cannot roughly divide the cells and put it over there in so many flasks. So we have to go for a counting. Counting can be done by the help of the special instrument which is called as Hemocytometer or Neubach chamber. So in the chamber will be look like all the four corners, it is having the 16 boxes. To the 16 boxes, we have to load the cells and observe how many live cells are there, how many dead cells are there. So this can be done with the help of the another one more solution which is called as trypan blue. The function of the trypan blue is it will go and attached, or it will form the, it will go and uptake wherever the dead cells are there, it will be uptaken by this particular drug. So this trypsin which will be taken by the particular cells, and they will have a shrunken appearance, but in the case of the live cells they have a gloomy appearance and they will be presented over there. They will never uptake that blue color solution. So we have to count that. It is easy to count it over there on the boxes. The cells which are present inside the boxes only we have to count it over there, and the cells which are touched on the walls, which we should never ever count it over there. So inside the boxes, and number of cells more present on the one place, like 8 cells are in one place, we can calculate. So number of cells present on the boxes we can easily calculate over there. And we have a 4 boxes I told you, on the top 2 and bottom 2. So we have to name it as A, B, C, D. So all the 4 corners we have to count the cells. Live cells also counted, dead cells also counted, and there is one formula which has to be adapted over there to count the cell viable cells. So how to calculate the percentage of viable cells means that is equal to number of dead cells divided by total number of cells into 100. So we can easily get the cell viable cells. From that we have to be dilute the cells, and we have to transform into the different flask, and we have to utilize for our purposes. So if you are keeping that inside the incubator for about another 1 more day, it will have a full monolayer, and that should be again which will be utilized for the students. So if it is you are having bulky number of the cells in your hand, we have to go for a freezing technology. So this is what the maintenance and preserving the cells. So whenever we want cells, so you already have preserved most of the cells, but we needed another 1 more cell. So we have to take it out from this one. That is called as we have to take it out. Preserved cells has to be taken out from this one, from this particular liquid nitrogen can. That is called as revival of the cells. So we can revive the cells. So that can be done by the help of the taking out the cells, and we have to go for a centrifugation. Remove the dead cells. Regularly we have a 10% of the dead cells will be presented over there. Remove the dead cells, and then we have to go for centrifugation with again the media, and we will be having a gloomy appearance of the cells. So that has to be transferred into the flask, and add some amount of some 20% of the growth media has to be added over there, and wait for about 3 days by removing the supernatant or by removing the tissue culture fluid, and we have to be getting a full monolayer on the 5th or 6th day with the addition of all this content. So this is what the regular methodology which has to be adapted for a cell culturing and maintenance, and also preserving, and also we are going for a revival. Whenever we want, we have to go for a revival technique. So these are all the techniques are adapted for a culturing of a particular cell lines in one particular laboratory. Next we have to see about the toxicity assay. This is the major assay. This is the one which will be all the pharma people will be utilizing this particular technique for a simple drug which they are invented. So that has to be finally, it has to be tested. So nothing but the name itself, it is giving the information that cell toxicity assay, nothing but the particular drug is toxic to the cells or not, that has to be evaluated. So for this, first we have to grow the cells in the 96 well plate, and we have to also add the different concentrations of our drug into this one in triplicate. Generally we usually used to add a triplicates, so different concentration like 5 mg, 2.5 mg, we can go for a double dilution of the particular drug, and we have to add it over there in a triplicate wells, and then we have to incubate for about 1 full day. So why we are incubating means the cell already it was 96 well plate, it was coated over there on the first day. Actually this full work is 3-day work. So how to perform means first day we have to load the full cells 96 well using a 96 well plate, we have to load the cells or seeding the cells on the very first day. Then followed by we have to wait for 1 full day to get a monolayer of the particular cells on the 96 well plate. Second day we have to add different concentrations of our drug. For example 5 mg, 2.5 mg, like that we have to add 2, 4, like that we have to get ready with that one. So depending upon the concentration we have to prepare the triplicate wells, and we have to load the particular drugs into these particular cells, and again we have to leave it for 1 more day. Why we are keeping for 1 more day means the drug has to show its action. So both of them have to have a competition has to be happened over there, and it has to show its pharmacodynamic and kinetic effects over there, followed by it has to be whether it is die, depending upon the concentration the cell will die or the drug will be having their actions or not, that has to be evaluated over there. So if you are putting a different concentration, so the cells which will be compete with each cell and the drug will be compete with each other. So for that we are leaving for about 2nd day. On the 3rd day we remove all the content. and we have to add about MTT 20 microliter, and we have to incubate again for 4 to 6 hours. So why we are adding means the fine function of this MTT, it has to go and add it over there wherever the live mitochondrial dehydrogenase is present inside the cells. So the live cells only possess this one. So wherever it is there, it will go and attach over there and form there is one complex. The MTT, which is named it as 3,4,5-dimethyl thiazole 2-YL 2,5-diphenyl tetrasodium bromide. So this will form a complex. So this MTT, which will go and join to the live mitochondrial dehydrogenase, and they form a formosome crystals, which is not a soluble one, so that will be formed it over there in that place. So that has to be neutralized by the addition of adding some amount of DMSO, and then after that we have to go and take the reading in the ELISA reader, and we have to calculate how much amount of the live cells, how much of the dead cells, and through this calculation we have to run a graph, put a graph, and we have to find out how many cells are having the 50% of the confluence in that area. So that should be assessed depending upon their concentration, and we have to evaluate this one. This is called as MTT assay. Now what all the applications of performing these work. So the fine application which is started with the major development is, first we will have a antibiotic emergence. Antibiotics, they prepared the different types of antibiotics. Major development of this particular technique is, number 1 is production of antibiotics, number 2 is production of different varieties of enzymes like trypsinase, collagenase, which is useful for their cleaving of the particular tissue matrix into the respective cells. And the third one is the production of different types of mediums. So that has to be used for the culturing of the particular cells. So all these are the major developments which are occurred by this period. Application, as for the application part, it is utilized in the number 1 toxicity panels. So most of the people, they will be inventing different types of drug, or different types of the varieties of nanoparticles, or different types of the plant extracts. So that has to be easily tested whether it is toxic to the particular cells or not. So this will be preliminary, they will be doing it in the in vitro assay, then they can go for in vivo assay, or else they will go and kill the animal. So that will create some ethical issues. To prevent that, we will go for these different types of methodologies. Number 2 is, it is also used as a model system. So if you are doing this, if you are getting this particular cells and their morphology and the structure analysis, it is very much useful to know the developmental stages of the varieties of the cells, and their functions has to be easily evaluated. Playing the major role in the cancer research. How means, we have to particular cancer cell lines in our hand, we have to put a different drugs which we are synthesized over there. It has to be put it over there and see to that whether this drug is having their actions to kill this cancer cells or not. That has to be done easily. So that is what it is. It is having the major application in the cancer research. And then virology. This is an important one because we have a varieties of vaccines. So those vaccines are produced using this virological techniques. So how means, we have to cultivate varieties of viruses using these material as a nutrient material, and the virus will go and it will form a variety, it will grow, which it will be utilizing the material for their system, and they will start grow or multiplying more and more, and it has to be taken it out, and we have to go and produce the different types of vaccines by inactivating this particular viruses. So that is the major work of the, and then virology concern. Gene therapy method, so there will be a replacement of the genes, can also be adopted using this particular cell culture techniques. So, and also we are having a genetic engineering techniques. In the techniques, we are preparing the varieties of protein by producing the monoclonal antibodies, but by incorporating the varieties of the proteins into this one, and we are doing this work. So all these are the major applications of this cell culture. Till now I explained about you the basics of the cell culture techniques. In future I will be coming with some other important techniques which are used for our human health and diseases. Thank you very much.